Hello to the people that are joining us. We'll wait a couple of minutes and we'll get started.
Hi.
Hi, Shubha.
Hi, Shubha.
Patricia. Oh, Nandini without the camera.
Yeah.
All set, Ajit?
I think so, or at least as set as I'll be.
You said you wanted to talk to me, Ajit, when we met last, but I don't know if you tried to get in touch with me after that, and I may have missed you.
No, I kind of got drowned in stuff as soon as I came back, Shubha, but I will text you and then set up a time.
Okay. Thank you for doing this, Ajit.
My pleasure. Hopefully it works, as in it's interesting, but this is definitely what I've been doing the last couple of years. It's I think all of you are very familiar, in the sense that this is potentially maybe a little bit more of a discovery for me, but community-based work is more complicated than, or takes more time than normal science I do.
True. Yeah. Takes perseverance. You may be finding.
Right. It took 4 tries of proposals to NSF before it got funded. And sometimes it's tempting to just give up and say, okay, this is not working, or do it differently. But in this case, I think it is worth it.
Great, that succeeded.
Mm.
Okay, I think that we can get started. 3 minutes have already passed from the time. So, good morning, good afternoon, or good evening, depending on your time zone. Welcome to this new webinar of the Trevor Platt Science Foundation. The foundation, as you know, is an international organization dedicated to capacity building in science, research, and education related to our oceans, working towards stewardship of our planet for future generations. Today's webinar is one of the capacity building activities of the foundation. So before we get started, just a couple of comments. As you can see, your microphones are off, except for the panelists. And please post your questions in the Q&A box, and we'll get to them at the end of the webinar. So today is my pleasure to introduce our colleague and friend, Ajit Subramaniam. He's a Lamont Research Professor at the Lamont Doherty Earth Observatory of Columbia University, and is an oceanographer with expertise in biochemical cycles, remote sensing, biooptics, and phytoplankton physiology. He is interested in advancing our ability to observe the ocean and expand our understanding how the marine ecosystem works and can be managed. Ajit earned his PhD in coastal oceanography and MS in marine environmental science from sunny Stony Brook. He has a bachelor's degree in physics from the American College in India. And he will talk us about co-production of knowledge and an emerging threat of CyanoHABs in the Arctic. So, Ajit, the floor is yours.
Thank you, Vivian. Thank you for inviting me to do this. I have been doing this for a while, and quite excited to share some of its ongoing work. So sort of share with you the motivations for what we are doing and what we are finding along the way. So this opening slide basically shows my colleagues at the Native Village of Kotzebue, who are basically co-PIs on the project that I'm working on, along with the mooring that we've used there. So to locate you, we are talking about a community in Northwest Alaska. So there you're seeing a map of Alaska. And essentially, if you zoom in a little bit, you're seeing a place where we have a few different rivers come in. So this water body here is Kotzebue Sound. And you have the Noatak River that comes in there. We have the Kobuk River Delta that we can see here. And just off to the east here is the Selawik River. And all three of them basically flow through this one pinch point. And this bright spot that you see here is the Native Village of Kotzebue. They're also shown on this map are some of the sampling locations that we go to. And the red star there is the place where we have our mooring that we put out during the ice-free season, basically from end of June, early July through end of September. But to begin the story where it actually begins, this is back in 2008 when someone noticed, some members of the community noticed this kind of a green thing in the water. They didn't know what it was, and they brought it to the attention of the environmental director for the Native Village of Kotzebue, saying that perhaps someone had spilled paint in the water because it had for them a very fluorescent green color. They took some samples and sent it off for analysis, and thinking it was paint to figure out where it might come from, and it was not paint. And in the meantime, they found that there was sort of the scum along the beaches that had this sort of in this case, it's a little bit of a faded cyan color. And in the end, they've basically sampled for all the chemicals and didn't find it there. But they also found this bloom in the middle of Kobuk Lake, which I'll show you later in the slides. And they took the water samples and had them analyzed and basically figured out that it was blooms of cyanobacteria. And they also had analyzed it for toxins and found measurable quantities of microcystin and nodularin, both of which are cyanotoxins that affect the liver. Since 2008 and 2009, these have been noted by the environmental director of the Native Village of Kotzebue in his weekly journal as happening sometime in late August, early September, almost every year. So basically, we have a sense that something might have changed. And so this is a photograph, and here you can see the blue color much more distinctly of the bathtub ring, as we call it, that was left behind at the end of bloom of the cyanobacteria. And this actually was from July. And I'll get back to the significance of the idea that the original blooms were noticed in September, early September or late August, and the fact that we actually found it in July, which was a big surprise for us. It shouldn't have been, but it was. But then in August as well, these are photographs taken from the air and from the boat. We see these blooms in the water. So the question is, why and where did this come from? So just a very quick little bit of information on the Arctic, which most of you probably know, but if you think of the daily average sea ice fraction of the Arctic, then basically what we are seeing is the decreasing ice cover. And the decrease in ice cover, and this is for September, so you can see there's a decreasing ice cover both in March and September, but the trend into September is much higher. And this has consequences to the ecology, the biogeochemistry and the ecology of the system. And so just to quickly think about that, what we have had in the system so far is that we basically have the sun coming, making an appearance in March, April. So if you think of it as a phenology, you have the sun making an appearance in March, April, but the water is still covered with sea ice. And sometime in May, you basically can have the development of strong blooms of sea ice algae that fall into the water column and are an extremely important source for both the benthic community as well as the walruses and seals and things like that that use the benthic consume the benthic community, and the whales. The whales as well. Yes, whales, walruses, and seals are an important part of the food source for the human beings, the Inupiat community that live around there. So what we have right now is a phenology where you have the sun basically increasing, sea ice decreasing as we go in time. Then you basically move from a sea ice algal community to a pelagic phytoplankton community. And then by the time you come to September, you start growing the sea ice again, and you then run out of light, you run out of nutrients, and most of the phytoplankton go away. So this is the phenology that has existed in the past. What we are now starting to see is a phenology where the sea ice disappears much earlier, which basically means the pelagic phytoplankton community comes in a lot earlier, which means the nutrients get used up earlier, and the sea ice formation in the fall is delayed, which basically means that we now also are seeing the increase in fall blooms, which was not something that was noticed before. And again, by the time we finish, hopefully the significance of that will come clear. So this is basically the change in the phenology that has been happening. Again, perhaps something to note here is the idea that you are having the phytoplankton blooms happening earlier, and thereby depleting the water of nutrients by the time you come into the late summer timeframe, which has then consequences to what kind of phytoplankton will grow. This change in phenology is critical in a whole bunch of different ways. I'm approaching this with a very phytoplankton-centric view because that's what I study. But do remember that this then has impact on the higher up in the food web, and eventually on human needs for food. As I will mention, well, the community around Kotzebue, let's see, we'll get there in a second. So basically, we are talking about phytoplankton, feeding the zooplankton, feeding the benthic shellfish and crustaceans, feeding small fish, which then feed the larger fish, marine mammals. And while the phytoplankton are critical for for supporting the system, it also has to be remembered that not all phytoplankton are good. And basically, one of the things that has come of great concern is that this change in phenology and warming of the waters is also giving rise to harmful algal blooms that have not been seen there before. Some of the blooms are acutely toxic, where you can the person can be killed by ingestion of a few cells. Some of the blooms, such as the cyanotoxins that we are studying, are chronic, as in they are hepatotoxins. They affect your liver, which means you're not going to keel over and die tomorrow. But on the other hand, you'll have a slow deterioration of your liver function, which basically has a end mortality effect. And then there are other harmful algal blooms where it may they not be toxic, but they still kill, or they still have nuisance effects, either by way of creating hypoxia or killing fish in a different mode than toxicity. And hopefully we'll have time to touch on that. But the net result is that you have an ecosystem that is now being impacted by climate change, where we are seeing, on the one hand, an increase in productivity because you have the longer growth period in the Arctic. But on the other hand, you also have deleterious effects, such as harmful algal blooms being formed. So what we've been focusing on are cyanobacterial blooms in this system. And here is a photograph taken through a microscope of Dolichospermum. filaments. And one of the things you'll see here, and we'll see this in some of the other photographs as well, is the presence of heterocysts, such as that one you see there. What you're seeing here are individual cells that form long chains. And in the water, they may look like grass clippings or something like that. So these are basically the cyanobacteria that is approaching at the surface of the water. Just as a refresher, a few points on what cyanobacteria are. So the word comes from a Greek word, cyan, which is blue. These are gram-negative bacteria. It's important to note they are bacteria. They are ancient, but they do photosynthesis, and therefore sometimes they are phytoplankton. They are seen as they are called algae, and because of the color of the freshwater cyanobacteria, they're called blue-green algae. They have basically we've seen them from 2 billion years ago. They are thought to be partly responsible for the oxygenation of the Earth's atmosphere. But then they also have certain genes or enzymes in them, nitrogenase, which allows them to fix nitrogen, but are poisoned by oxygen. So their evolutionary traits actually come through in a few different ways. They predate the existence of the present ozone shield, and there is a consequence to that, which is that they actually have sheath pigments which give them UV protection, which are not found in all phytoplankton. Let's see. They make a contribution to the primary, so they are the most numerous phytoplankton in terms of numbers. They make a significant contribution to primary production in the ocean, but they could be the dominant primary producers in extreme environments, such as in cold deserts, hot springs, hypersaline environments. They have an enormous total biomass for those who are interested in that kind of statistics. In the oceans, Prochlorococcus is the most numerous photosynthetic organism. My favorite beast there is Trichodesmium, which is something that I studied a long time ago. But when they have a funny contradictory presence in that they both are the smallest photosynthetic organism in terms of Prochlorococcus, but because some of those species form colonies, form filaments and colonies, they're also potentially large and even visible to the eye. So the blue-green algae cyanobacteria have this extreme existence in the size range. I think one of the figures I've seen in other people's talks is that basically you have the same range as the size of a fish to the size of Manhattan, sort of stretched in that sense of when you go from Prochlorococcus to something like Trichodesmium, which could be a couple of millimeters a whole colony. So you have essentially the same several orders of magnitude range in sizes. There are several of them produce toxins, and one thing that is important to remember is that while we as human beings think of them as toxin producers for reasons such as allopathy, to prevent them from being grazed and things like that, we do have to remember that it is entirely possible that the toxins that they produce are chemicals that also allow them to get rid of free radicals within the cell. So it might have nothing to do with the toxic effect as much as we understand it, as much as it has something that it has a physiological function. They're producers of hepatotoxins, which basically affects the liver. Some of them can fix nitrogen, which makes them really important in the global nitrogen cycle, and also plays an important role in how we can control them for example, in freshwater systems, where instead of it being a nitrogen control system, you'll have to control it as a phosphate control system because that's the other element the other nutrient that they need. They have chlorophyll as their major photosynthetic pigment, but They also carry, what is unique to them is they also carry phycobilins as light harvesting pigments. And this, again, the reason I'm mentioning these things is because these have effects on how we can see them optically or with remote sensing. They have gas vesicles, which allows them to migrate vertically in the water column, which again is an important thing to remember, which is if you think of this as, for example, in a freshwater system such as a pond, where light is as an important limiting limits growth as nutrients do, being able to move up and down the water column gives them an ability to basically outcompete some of the other phytoplankton. But the gas vesicles, again, have an optical component which helps us identify them because they scatter light. The fact that they can vertically migrate has other optical implications, which is that they can live very deep in the water column, like in the open ocean. They could be a major component of the deep chlorophyll max, where the penetrating light is mostly blue. Or in a system like in a lake, they could be found very close to the near the surface, but still, because of the presence of CDOM and detrital matter, your light quality shifts more to the green or to the yellow range of the spectrum. So the fact that they're able to have gas vesicles and they have phycobilin proteins gives them a unique ability to maximize photosynthesis in these very variable conditions. They are filamentous, as I had mentioned, which means that they can actually form colonies that we can see with our naked eye. Sort of to drive home this idea that these are found in very turbid waters. Again, here's a satellite image of the study region, and you can see the amount of sediments that's coming out of the Noatak River, as well as the amount of dissolved organic matter that comes out of the Kobuk River. And this is basically the region where we are finding a lot of cyanobacteria. So sort of, if you go back to how the kind of waters that they live in, we made measurements of the absorption spectrum, and we basically see that CDOM and detritus contribute almost 90% of the absorption in the blue wavelengths, and phytoplankton only have about 10% of the absorption. Whereas when you get into the more red wavelengths, phytoplankton actually have a little bit more than 50% of the absorption. So given this is the light conditions they are found in, they have adapted their photosynthetic pigment capacity to do photosynthesis accordingly. So the primary the receptor pigments that you find in cyanobacteria are phycoerythrin, phycocyanin, allophycocyanin, that then feeds into photosystem 1 and photosystem 2. Perhaps the easy way to sort of think about this is essentially the path of a photon that goes into providing photosynthesis. Or if you think of it spectrally, you're absorbing light 495, 570 nanometers, and then to moving it to the phycocyanin, then to allophycocyanin, to chlorophyll, and to the photosystem. So this sort of a progression in the photosynthesis capacity is a response to the amount of light that's available at the various wavelengths. So if you look at Trichodesmium, which is a marine cyanobacteria that is found in the open ocean, where you don't have a problem with CDOM and absorption in the blue, you look at the fluorescent spectra there, when you excite at 495, you basically see the response at 570, and then you see how the other fluorescence peaks are seen for phycocyanin, allophycocyanin, and to chlorophyll, and to photosystem 1. So what this is all means in terms of phytoplankton in general is that most phytoplankton, you can see the chlorophyll absorption at 443 nanometers and at 675 nanometers, whereas with something like a cyanobacteria like Trichodesmium, you have additional absorption at 495 and 565 and 620 nanometers that basically are due to the phycobilin proteins and help with the photosynthetic capacity of the cyanobacteria. in terms of actual data, in terms of the waters that we're seeing around Kotzebue, you can see that the absorption due to CDOM has a spectral shape that looks like this. Oops. The absorption due to particles, total particles, has an absorption shape which is also very high in the blue, with a small bump that you see at 695 that is due to chlorophyll. But bulk of this absorption is coming from the non-algal pigments or non-algal absorption, which is due to the sediments that we see that's coming out of the rivers. So in terms of the phytoplankton due to the absorption due to phytoplankton, you can see that it's almost an order of magnitude lower than the total absorption that we see for particles. And this is essentially what we are finding in the waters around Kotzebue. So we can see the chlorophyll absorption, but we can also see the phycoerythrin and phycocyanin absorption in these waters. The third thing is the absorption due to mycosporine-like amino acids. And this is essentially found as a sheath pigment in a lot of cyanobacteria as a way to protect them from UV damage. So as I mentioned, cyanobacteria existed before there was an ozone in the atmosphere to protect against UV. And so they are already developed evolutionarily mechanisms to protect the DNA from UV damage. And mycosporine-like amino acids is one of the ways in which they do that. What do they look like? So if you look under a microscope, you might see filamentous colonies like this. This is Aphanizomenon, one of the colony-forming cyanobacteria that we see around in Kotzebue. If you look at it, if you take a bucket of seawater in a bloom or water in a bloom, you might see something that looks like this, where you see, like I said, the colonies are visible to the eye. So these are basically colonies of cyanobacteria that we see in the water. If you look through the porthole of a boat underwater, you might see something that looks like this. This is a photograph taken in the Baltic Sea, where you have blooms of cyanobacteria of Nodularia, Aphanizomenon happening most of the summers. So this is what it looks like when you look at it in the water. If you look at it over the side, you might see something like this. And one of the reasons I like this picture is that not only can you see the blooms of cyanobacteria, but you're also seeing sky glint, which is one of the clues that we have to remote sensing of this organism, because what you can see is that the cyanobacteria are very close to the surface, but they don't actually break the surface. And you can also see that in this blue photograph of a trichodesmium bloom in the South Pacific, where you can see that there is sky glint is visible where you have cyanobacteria in the water, whereas something like this, which is a piece of pumice that, because there's an underwater volcano that had erupted nearby, this is a piece of pumice that has come out, and so you can see that there is no sky glint associated with that. So the idea is that you can see things that are close to the surface that is still affected by water properties, and then things that are above the surface which are not. So this is an important facet to how we are able to remote sense this blooms of cyanobacteria. This is another picture just to show you how complicated it can be if you actually want to develop algorithms and validate any kind of quantification of cyanobacteria. Because when you have patchy systems like this, the question is, are you looking at this place or are you looking at this place? Where do you take your water sample from? How do you count the cells, or how do you take chlorophyll or phycocyanin concentration measurements? And what does it all mean when you're looking at it with a satellite that has a 1 kilometer spatial resolution? So cyanobacteria are exciting. I don't know that I don't have confidence that I'm going to have a ability to do much with them before my time has passed, because they've been around for over 3 billion years and they seem to know what they're doing in the waters out there, but they are intriguing in a way that challenges us, and I really like working with them. This is what they look like if you see from an airplane, and here's a satellite image of the cyanobacteria blooms in the Baltic. This is Baltic proper, Sweden is off here, and Poland and Germany are down here. And you can see that they can be very extensive in places like in the Baltic Sea. As much as we know about cyanobacteria, because they're common in freshwater lakes, in water reservoirs, where they actually have a huge impact on human beings because of the fact that they carry these hepatotoxins, the fact that they've been studied in the Baltic for a long time, all of that still makes it surprising that they appeared north of the Arctic Circle, where we found them. One last thing about remote sensing, because that's what I do, is here is a Landsat image of a Trichodesmium bloom in northern Australia. And if you basically use our standard algorithms, you basically see that although there's a very clear image, a lot of this basically is getting masked out as clouds. And that's because the very near surface or surface blooms of cyanobacteria have a high reflectance in the near IR bands, which have been traditionally used for cloud masking. We have moved in many satellites to shortwave infrared bands, but the fact is that in the near IR bands, the fact that they have a reflectance used to be how the dark pixel assumption used to work, and they got masked out. However, if you look at them in a coarser satellite image, so there's a satellite image, there's a MODIS image from the same day, near the same time, you see that the clouds go away. And that's because when you homogenize over a kilometer, you don't have the same effect because you're now mixing a surface bloom with water next to it. If you remember how patchy it is, you're mixing surface blooms with water. And so over a 1 kilometer pixel, you don't have the same high reflectance in the near IR as you do when you're actually looking at it in extremely high resolution imagery. So there are complications when you go high resolution because you're starting to see features that might basically be averaged out in a coarser resolution. All of these basic for us present both an opportunity and challenge in terms of how do we detect and quantify cyanobacteria from space. Back to our study site. So here is a Landsat image of a cyanobacteria bloom from September of 2026. So you can see extensive regions of cyanobacteria that have accumulated at the surface that also show some of the beautiful water circulation patterns. And that's essentially what you're seeing here, some sort of eddy features. My colleague in Kotzebue, you'll see it in a second, so I'll hold back on that. These are some of our, this is the Native Village of Kotzebue. Actually, that white line there is the runway, is the airport runway. So you can see it's a fairly small area. It has about 4,000 people. The only way to get to Kotzebue is either by airplane or by boats in the summer when there's no ice around here. But the Kotzebue Sound itself is very shallow. It's often, in most places, it's less than 5 meters deep. And so you can't get big ships in there. It's small, shallow barges that are able to come in. The point being that this is a remote community that is cut off. And for food, the people here depend on what they can gather. Subsistence is the major source of food because it is really expensive to otherwise bring in food by airplane, which is the only other way to bring food in here. So because so much of their food intake depends on the waters around, the idea that you have cyanobacteria blooms and some of them are toxic could essentially be, is a matter of grave concern. And I was there on a different project where we were working with the community on the effects of the change in sea ice cover. And when I was talking to Alex, my colleague Alex Whiting, who's the director of the Native Village of Kotzebue's environmental program, he was talking about the concern that they have for cyanobacteria. And because I'd worked on cyanobacteria, it was a good match. And we started focusing on trying to understand where we are finding the cyanobacteria, when we are finding the cyanobacteria, and why they are there, what's causing them, and the implications to the ecosystem and potential food safety. So what is their toxicity? One of the one of the peculiar things about cyanobacterial toxicity is that not all cyanobacteria are toxic, and they're not toxic all the time. So one of the questions we are asking is also what's inducing toxicity? Are we looking at impacts of temperature stress, nutrient stress, so on and so forth? And that again is thinking about it very anthropomorphically. The idea that they're stressed to produce toxins may or may not be the right way to frame the problem. It might be a physiological response that we don't understand, but that's the kind of thing we're interested in following up in this work that we're doing there. So again, to remind you, the Native Village of Kotzebue is there. It's about 35 miles north of the Arctic Circle. It's in the northwest corner of Alaska, just past the Bering Straits. We have 3 major rivers that come out here. And so that's essentially the impact that we're seeing. And one of the other questions is, are the cyanobacteria that we're seeing in Kotzebue growing locally, or are they basically freshwater species that are actually growing in the rivers and then being advected around where they're seen in Kotzebue? The native village of Kotzebue has a population of about 4,000 people, and it is the largest community in the northwest in the northwest of Alaska. It's where the Northwest Borough has its capital, if you will. There are much smaller communities that are distributed around, and I mentioned that because the fact that they were seen in 2008 doesn't necessarily mean that they didn't happen before. It is such a sparsely populated region that it is entirely possible that there were cyanobacteria there and nobody noticed it, or it is a new phenomenon. And this is one of the things we are trying to understand. One of the, then again, as a part of the project, we deploy a surface mooring at this location where the Three River, where we hope to capture the water from the Three Rivers. We put this out in the end of June or early July when the ice is blown out. The whole region is ice covered till about the end of June. And so we wait for the ice to go away before we put in the mooring. And then we leave the mooring in till the end of September, when ice starts forming again, or just before ice starts forming again. We pull out the mooring. So we basically have data from the mooring for the ice-free season. And this is, so I'm going to show you some typical data from 3 different years. There were a few surprises for us. One was, oh, I'm so sorry. They're doing construction next door. I told them to stop, but hopefully it's not going to disturb us too much. One of the surprises for us was this spikes in high salinity that we saw in the summer of 22. This was both a surprise because we didn't know, because normally the salinity is basically between 0 and 5. It's mostly a freshwater system. But the idea that there are high salinity waters that is coming in from the Chukchi Sea at the surface, these are surface salinity measurements that we are talking about here, was of enormous concern because, as I think when I finish up, there's another harmful algal bloom, toxic bloom of Alexandrium that forms just outside the mouth of Kotzebue Sound, in the Chukchi Sea, just outside Kotzebue Sound. Alexandrium is basically, it's thought not to do well in salinities less than about 18 parts per thousand, and therefore the idea was that the waters around Kotzebue would not be as affected by Alexandrium as the communities that are facing the Chukchi Sea directly. But the fact that we are actually seeing high salinity waters coming into Kotzebue, coming by Kotzebue, now changes that understanding a little bit, which is that there is a potential for the Alexandrium to have impacts on the food, the fish, the shellfish that are harvested near Kotzebue. The other surprise was, well, maybe it's not so much of a surprise, but you can see that the highest temperatures are found in the July timeframe. And by the time we get to mid-August and September, the temperature starts to fall off. In places like the Baltic, the cyanobacteria blooms have been noticed to happen at the end of summer when there's a whole story of it's the warmest, the waters are nutrient depleted, and then you have cyanobacteria that can fix nitrogen that can then grow there. Out here, the fact that the warmest waters are found in at the beginning of summer rather than end of summer was also something that we didn't anticipate, because the cyanobacteria blooms that have been visually noticed have always happened in the mid-August through early September timeframe. Then we put out the mooring again in 23, and you can see here we had 5 different spikes of high salinity. This one is slightly different. This spike is due to an extratropical system that went over, and it actually created enormous damage in a community south of Kotzebue in Nome, where entire houses were lifted off their foundations and floated out into the water. Extreme events of rainfall and flooding and storms have become more common now with the disappearance of sea ice. But that's what that is due to. But we see 5 other incidents of high salinity spikes that happen in the middle of summer. Whereas when you looked at 23, we found only 1 real spike. So one of the things we are finding here is that there is a really big intraannual difference in the for waters from the Chukchi coming in. The traditional knowledge about this has been that if you have winds out of the west, you basically are pushing Chukchi Sea water into Kotzebue Sound and into Kobuk Lake, and that's essentially when you see these high salinity events. Was the hypothesis, was the thought. But we did extensive analysis of wind speed and direction from the instrument that is based at the Kotzebue airport, and found that it was actually not so simple. There was no simple relationship between wind direction or wind speed and when we were seeing these high salinity events. And then in the summer of 24, we had no high salinity events at all. So the idea that there's this huge intraannual variability is also point of great humility for me, which is that, you just can't go and make one set of measurements and say, you know how the system works. In a system like this, nature always seems to have different ways of surprising you. So just as to put it all together, you see a lot of spikes in 22, only one spike in 23, and no spikes at all in 24. We don't have good river discharge data for the three rivers. Only the Kobuk actually has a discharge station that is way upriver. The NVOK doesn't have any, and the Salavik doesn't have any. But what we did do was to look at what the discharge was in the one station that we have in the Kobuk, and found that what we found was that there was a period of low discharge through most of the summer of 22. And so the idea is now that it's a combination of winds and discharge, and potentially some tidal influence as well, though tides are not very big here, that allowed for these big spikes in high salinity events coming into Kotzebue, whereas in 23, we had mostly much higher discharge. So that basically prevented the discharge of the river, basically pushed the high salinity water, kept it away. So we didn't have these sorts of spikes that we saw in the summer of 22. Just as some of the other measurements we've been doing there is going to the different locations and doing measurements as and when the members of the community can do it, and just trying to find some patterns in the seasonal changes in terms of temperature at the different locations, as well as looking at what the nutrient concentrations are at the different locations. I put this up here because one of the things that many cyanobacteria can do, not all, many cyanobacteria can do, is they fix nitrogen, and therefore it doesn't matter how much nitrogen there is in the system, but they can fix their own nitrogen if there's not enough. But they also have the ability to take up dissolved organic phosphorus, which many of the other phytoplankton don't do. So the fact that we didn't find any measurable inorganic phosphorus, but we found reasonable numbers for dissolved organic phosphorus, all of which is coming out of the thawing of the permafrost, sort of sets up another hypothesis, which is that the cyanobacteria blooms that we are seeing happening in Kotzebue, that have been observed at least since 2008, are a consequence of the thawing of the permafrost that's mobilizing nutrients. It mobilizes not just the dissolved organic nutrients, but it's also mobilizing things like iron, which the nitrogen-fixing cyanobacteria need. So there's an entire different phenomenon called rusting of rivers, where the rivers are actually turning rust-colored because of the oxidation of iron that is being exposed as we have permafrost slumping into the water and exposing new permafrost and coming into the water. So you have mobilization of iron, you have dissolved organic phosphorus, and that basically provides a home for cyanobacteria in the recent past that they didn't used to have maybe a long time ago before the thawing started up in earnest. The typical phytoplankton that we see there are some diatoms. We have not actually seen many we have not seen Alexandrium catenella at the waters in Kotzebue itself, but they are found in extremely high concentrations just outside, and I'll show you that in a map in a little while. As well as we see cyanobacteria such as Aphanizomenon or Dolichospermum. This is perhaps not the best way to represent it. I'm still trying to figure out how to do this better, but this is counts of phytoplankton, microscopy counts of phytoplankton we're seeing there. The green squares, just to draw your attention, we're looking at the green squares in these locations and the not so clear yellow crosses at these plus signs at these locations. And for me, the intriguing thing is that if you're going to use a nutrient argument for why we have cyanobacteria, how do we explain the fact that we're actually seeing large numbers of diatoms at the same time at these locations? So for me, there are still some open questions in terms of how the system is working and what is controlling the growth of either cyanobacteria or other phytoplankton. And if indeed we are seeing any kind of a shift from diatoms to cyanobacteria, which was one of our initial hypotheses and concerns, which is that the food quality that's going to basically be available further up the food chain might be impacted if you're starting to replace diatoms by cyanobacteria. At this point, with the numbers that we have, it's not entirely clear that's the story, but it is something that we are sort of still poking at, trying to understand. We can see that the cyanobacteria, when we integrate them at different locations, don't have the same really strong seasonal cycle that we thought was there when we went in, because visual observations of cyanobacteria blooms in the waters around Kotzebue have always been end of August, early September, never earlier than that. And now we are seeing potentially that maybe there isn't such a strong seasonality here. These are, again, photographs of some of the typical cyanobacteria that we see there. The reason I put this up is because you can see that in this case, in the Dolichospermum, you can see heterosis, and we see heterosis in the Aphanizomenon as well, which basically says that they're actively fixing nitrogen. So this is a system where nitrogen fixation is required. But at the same time, we're seeing something like Astronella, a diatom, that is also blooming there in high numbers. So we are still trying to figure out how that works. And then, of course, what is also really intriguing is that we also see Dinobrion, which is a mixotroph in these waters. And we know even less about what Dinobrion is doing, whether it's actually exhibiting mixotrophy in terms of being taking up either consuming prey or taking up organic nutrients, or if it is being a regular phytoplankton and basically living as a phytoplankton. So there is interesting questions that still need to be followed up, that where phytoplankton are now starting to tell us something about the system, even though we are studying the phytoplankton because we want to understand how they impact humans. So just to go back, they produce cyanotoxins. They have the potential to decrease oxygen levels. And again, that's an important point that I will come back to. They might basically affect the food quality up the food chain and disrupt the ecosystem that way. It's important to note that unlike many other places, here's a place where 70% of the subsistence comes from waters around the village, and therefore even the smallest changes impact what people eat and how much they can eat. And the economic costs are also really serious problems. So these are all the reasons why the community is interested in doing this. And so we've been working with the community in terms of trying to understand these problems. One small aside as far as remote sensing goes is that so my colleague Alex calls this the smiley face, because almost every year we see blooms along that front here, of a front of some sort here. And we used the floating algal index, and we used Landsat and Sentinel-2 data, and used the floating algal index that we can produce using Acolyte to try and quantify the cyanobacterial blooms and see if we can say anything about change over time. The reasons for doing this, well, we'll get to it. So these are sort of the number of blooms that we have seen with cyanobacteria over time. One of the concerns I had was whether these surface blooms are happening because of wind speeds. And in other words, we're able to see surface blooms when the winds are low, and if the winds are high, then we don't see the blooms. And that is related to the fact that the blooms have been seen by people when they accumulate at the surface, what I call my charismatic microflora. People can see it, and that's why we see that's when they were noticed in August and September, when the wind speeds are generally lower than in July, when they're still in the water, but people are not seeing it because it's all well mixed, because the winds are a little bit higher in July. The other challenge with using something like Acolyte and floating algal index is that when you have thin, low clouds, that also can cause some contamination or give you false positives for the FAI index. So So we try to correct for all of this using a model and using wind speeds and a model to try and predict when we are seeing the cyanobacteria blooms. And in general, came up with this idea that the cyanobacteria blooms are increasing in size. Do note that this is both governed by changes in cloudiness. In other words, we might be seeing more cyanobacteria because it's clearer, as opposed to there being more cyanobacteria in the water. And it also has to do with wind. So atmospheric conditions might impact this in ways that we don't fully understand. So this may or may not be right, but this is essentially what the satellite data is telling us right now. The other part of the toxic toxic harmful algal blooms that I was telling you about is the issue with Alexandrium. And this is work by Don Anderson and colleagues at Woods Hole, where they have mapped enormous cyst beds in the Chukchi and Bering Seas. And actually, this is an earlier image. The cyst bed in the Bering Seas is even bigger than what we're seeing here. And Don usually likes to put a map of the Gulf of Maine next to this to show how small the cyst beds and the Alexandrium problem is in Gulf of Maine compared to what we're seeing in Alaska. Just again to locate you, Kotzebue is right there. This is the mouth of the of Kotzebue Sound, which is here, and the Chukchi Sea. And the concerns we were talking about in terms of salinity was that if you had high salinity water coming in, that would basically bring in Alexandrium to the water, to the waters and affect the food web around Kotzebue. The hypothesis that Don basically has presented is that it's the warming of waters that is essentially both moving the cysts into the Chukchi and also allows for the resuspension and the growth of Alexandrium in warmer waters in the Chukchi Sea. The unfortunate part of, not unfortunate, the other part of it, or the thing that we need to be very careful about is that when this hits popular press, it sort of takes on a different tone. So you have useful information such as the fact that people are finding harmful algal blooms and therefore toxic blooms, and therefore we have to be careful in what we eat. But at the same time, you can also have some breathless takes on this, where you start causing panic. You know, when you start talking about California-sized seabeds of toxic algae creeping into the Arctic, this causes panic, and this is not perhaps the best way you want to talk to the people about something that is affecting the foods that they eat. Since they don't have that many alternatives, this is something that we have to be very careful about. So they've developed a system of communicating where they do advise the local health authorities about the presence of toxic algae when they see them, but it is a lot more nuanced communication than just putting it out there as a, we found 100,000 cells per milliliter or something like that. This, for me, gets back to the whole importance of a community-based observatory where the community is fully involved in the measurements being made and have a role in basically communicating that to the other parts, to the wider audience. So one of the questions is, do we have cyanobacteria going later? Do we have fall blooms of diatoms? And all of this comes critical because in 2021, in October of 2021, there was a massive herring mortality event in Kotzebue where thousands of dead herring washed up on the beaches. This is following the summer when all those headlines about the toxic Alexandrium outside Kotzebue Sound hit the press. So normally under these conditions, the dead fish are consumed either by human beings or fed to sled dogs. But given the fact that this could be toxic and potential food source was not essentially made use of in a place where food is an important commodity. And the fact that we were done some sampling, we had left some instruments there, gave the community a better understanding of what was in the water. And what we did find in the end was, it is not a complete story, but what we did find was that it is possible, we found pustules of diatoms in the gills of the fish. And so it is entirely possible that the fish basically swam into a fall bloom of diatoms, and that basically impacted the gills, and then they had other reasons for which they died. But it was the presence of diatoms in the water that might be the primary trigger for the fish kill, not the dinoflagellate Alexandrium, which killed them through toxicity, thereby changing the narrative on whether it is safe to eat or not. And all of this, so that's just a picture of Catasaurus, which is the frustules that were found in the gills of the fish. So all of this goes back to the really importance of working with communities and understanding, providing them the tools by which they can communicate and understand what's going on. This is just something that was sent around in Facebook. It's a massive pile of krill that was washed up on the beaches. So things like this happen. And we don't know why they happen, but these have consequences. If here the worry is what happens to the whale cohorts that would have normally consumed this krill, and that impact is going to be felt years later in terms of maybe a smaller whale population, which again is an important food source for the community there. So this is basically, these are the kinds of pictures that were sent around in terms of what they found washed up on the beaches there. We do not know what killed this. This is the importance of the work that the community is doing there. I just want to finish up with a few words on co-production of knowledge, which is that this for me is a really important way in which we work, which is that we don't, the way I see it is I, as a scientist, do not go there and tell them what to do. We work together with the people there to develop the question that is of importance to them, but we each bring in our own strengths in terms of what we can do. How we do the work, how we observe, how we interpret, so on and so forth. So for me, this concept of two-eyed seeing was one that I found really appealing in terms of there are strengths of seeing with each eye, but you're seeing even better when you see it with two eyes. And for me, reciprocity, mutual respect, and is important components of how we approach the work that we do. There are the communities there have come up with protocols, and we also have ran a workshop here at Columbia on good practices for community-based work, where they basically said nothing about us without us. So they really have to be a part of this part of the knowledge creation process. There is some shall we say. These are obvious, except that it's useful to just remember that this is how we want to do it and be intentional about what we do. There is some say different opinions, I guess is one way to put it, on whether indigenous knowledge needs to be validated by science or not. There are people who say that it's good to have oral traditions that have been passed on by generations to be validated by instrument measurements. And there are other people who say that that knowledge exists by itself and it doesn't require validation because that's based on generations of observation as opposed to 1 year of measurements that you make with an instrument. So, you can see that obvious strengths and weakness, or not weaknesses, but the strengths and challenges that we have with how we merge the two things together. A different way of thinking about this is that it's easy to get stereotypical about this, so I want to be very careful in saying that this is not how it always is, but it's a useful framework, which is that indigenous knowledge is often spatially local, but deep in time, whereas a lot of what we do might be spread over a large space, but very shallow in time. Just to remind myself, we've been looking at this cyanobacterial blooms in this region for the last 4 years, or trying to understand the ecosystem in some fashion for the last few years, whereas the community that has lived there for thousands of years have had several seasons of observing something. And therefore, to dismiss the oral traditions as not science is something that you've got to be very careful about, and something that definitely should not be done. A different way of thinking about it is that indigenous knowledge is holistic, is systemic. It basically involves system thinking, right? So they see something like this. There's a gorgeous picture that I saw in a presentation by Carolina Behe. Whereas as scientists, we basically specialize deep in one thing, but we sometimes have silos that do not allow us to integrate across the system. And so for me, one of the pleasures in working with the community in Kotzebue is being able to do that integration by talking to the elders there, even though I'm coming in with rather narrow views of what remote sensing can do or what optics can do or what nutrient measurement can tell us. So with that, I think I will stop and see if there are questions.
Thank you, Ajit, for this very interesting and deep presentation. We're running out of time. I hope, Patricia, I don't know how we can do with the time. Can we extend a few minutes more?
I can take a couple of the questions I see in the
Okay, great,
Q&A.
great. We have a few minutes. So I will check some questions in the Q&A box. And okay, there is one from Anchal Maria and says, how are researchers trying to determine when cyanobacteria produce toxins and what causes them to do so?
So we're doing some lab experiments here with organisms that have both been isolated from Kotzebue as well as with cultures that exist in culture collections and adjusting the temperatures. So trying to understand Q10, the temperature optimum for different cyanobacteria and measuring toxicity by basically stressing them at different temperature levels. And we're doing similar measurements with nutrient stress. But this is something that we're doing here, and that's the sort of the only way we know how to do it. So I'm absolutely open to other ideas on trying to understand what causes cyanobacteria to produce the toxins.
Yes, not a simple question. Yes. There is another question by Mohan Kumar Das and says, under future climate scenarios, which strategies most effectively reduce harmful algal blooms? And how can we integrate, how can integrated climate, oceanographic, and policy approaches improve their management?
I must confess that I probably have a very depressing answer for Mohan, which is that I'm not really sure that we are going to be able to reduce cyanobacteria blooms if the reason that they're there is an increase in temperature. In other words, unless we are able to reduce temperatures, I'm not seeing a path to reducing to harmful algal blooms that way. However, I think that the real important thing that we're trying to figure out here is how do you reduce harm, at least to human beings. And so the kinds of work we're doing there, which is monitoring the cyanobacteria or Alexandrium, looking at their toxicity and communicating it to the community on when it is safe or not safe when it is not safe to eat, or giving them advice on when they should be concerned about eating it, is essentially the management approach we're taking now. But I think I would want to reiterate that our understanding of at least cyanobacteria blooms there is still in the very nascent stage. And so, but it's possible that we might understand what's causing the blooms and we might be able to come up with a management solution. But at this point, we are just looking at trying to figure out where they are and how they are functioning.
Yes. I don't see, let me see if there are more questions in the, there is another one from Anchal Maria. So will there be any type of stress from other organisms which make them produce toxins?
Yes, I mean, but I just want to say that, it has been traditionally thought of allopathy as the cause for toxin production, which is that you're basically making yourself less attractive a food source. However, what some researchers have found is that we are finding toxins even in a culture when there isn't something else. So the idea that it's a potential physiological response is sort of how we're approaching it. As you can imagine, doing whole ecosystem measurements or work on toxicity is fairly complicated, and perhaps there are other people doing it. Toxins are not my thing, and therefore I'm trying to keep it simple.
Okay, yes. So there is another question by Khadija Overhire. What recommendations do you have on engaging effectively with communities and working together better, scientists and locals?
So I'm glad you asked that question. Maybe this is something that can be distributed through the TerraPlat Foundation web email listserv or something like that. We have there have been many documents produced on co-production of knowledge and how to work well with communities, including what we have, and I'm happy to share that. This was developed through a workshop where we had invited participants from Papua New Guinea, from Canada, from Alaska and other places, and also locally here, who have all been working in Madagascar, who've been working with communities, and we developed the document in collaboration. It was co-produced with them, if you will.
Great. Yes, I should mention that Khadija I think she is an expert in this subject of working with the different communities, and she actually had proposed a session for the Trevor Platt Science Foundation next meeting on this subject. So hopefully you can join us, Ajit.
I definitely hope to be there. Yes.
Yeah.
Khadija added, still learning.
Yeah, so to add on to that, Khadija is implementing an outreach project funded by Trevor Platt Science Foundation in Morocco and Australia.
Yes, yes, it would be good to get together. So she says thank you also. And I don't see more questions in the Q&A, but let's see if there is anyone that has more questions here. Anyone from the panel?
May I? Yeah. Okay. So
Nandini, yes,
thank
we
you for
see
the
you.
wonderful presentation. Yeah, I'm also happy to see myself after a long time. My computers have started working. Yeah, thank you for the wonderful presentation. One doubt that I would like to ask is that with this nitrogen fixing ability, have you noticed that cyanobacteria is able to support another bloom by a dinoflagellate?
Right. So that
Because
that is one of the hanging questions then, Nandini, which is, when I showed you, there's very low DIN, dissolved inorganic nitrogen in the water, and yet we are seeing cyanobacteria and diatoms coexisting. Nitrogen fixing diatoms suggesting that nitrogen is limiting, but then sorry, let me try that again. Nitrogen fixing cyanobacteria, which suggests nitrogen is limiting at the same time as diatoms. It's possible that there's a transference going on to be explored.
yeah, thank you. Because we have had one experience like that in the Gulf of Mannar when there was a Nodularia bloom. It extended and it was there for more than 2 weeks, which is very rare. But then we saw that in the phytoplankton, Oscillatoria was also increasing in concentration, which actually sustained the bloom, the dinoflagellate bloom.
Yeah. No, with Trichodesmium in the open ocean, we've sort of seen that occasionally. But in these freshwater systems, it is actually surprising that DIN was as low as it was and DIP is as low as it was. And so it's a little bit more intriguing as to whether that kind of a support is happening. I'll have one of the students working with me is going to be there this summer, where we actually hope to take some genomic and transcriptomic samples to actually see if we are seeing upregulation of some genes that might give us some clue to this. Which obviously then has the policy implications that Angel and Mohan were asking about. So, if you want to control them, you've got to understand what's causing them. And so we really need to get to the bottom of that.
Yeah, it's interesting how the phytoplankton community develops, because as you say, you have a lot of cyanobacteria and at the same time diatoms. That's quite interesting, yes. Well, I will have myself a few questions, but maybe there are more specifics about the cyanobacteria. And I was wondering, you show a graph where you show the salinity peaks and the temperature progression. And in the lower panel, there was a chlorophyll fluorescence. And I was wondering, because we know that cyanobacteria do not fluoresce too much with chlorophyll. So, but in other graphs, you show number of cells, which is more, actually is more strict than that. So how do you handle the chlorophyll fluorescence, or do you use phycobilin fluorescence? I don't know.
Yeah, no. So the sonde that we use also has phycocyanin fluorescence measurements.
Okay.
It's just the phycocyanin fluorescence is really noisy, and as you know, chlorophyll fluorescence itself is kind of noisy. Phycocyanin is even noisier, so it's a little tricky. But I will say that I learned a lesson, which is that we were seeing some high phycocyanin in July, soon after we put out the mooring. And I was thinking that that can't be, because the idea has always been, based on the observations of people there, that the cyanobacteria come in at the end of summer. And therefore, seeing them in the middle of July was like, I'm not sure about this, until we have these photographs of the cyanobacteria blooms in the beginning of July that was taken actually by a high school student from there who worked with me. She did sampling through the rest of the year. So the school began in September. So she actually sampled once a month from September through May. And and she continued doing the work in June and July because I asked her to. And she took those photographs in July, where we found cyanobacteria in early July. And then it was like, oh, wait a second. Temperature is warmer in July than it is in September. So there's no reason why you can't have cyanobacteria there. So that sort of changed our thinking a little bit on this. Now I have to go back and say that, okay, maybe the phycocyanin fluorescence measurements are not wrong as I'd originally thought. Maybe they're real. But we're also doing extracted chlorophylls. We haven't done it as in a sustained way in the past, but we are starting to do that. So we'll have some sense of it.
Okay, great. So I don't know if there are more questions. Let me see. Oh, sorry. I keep on pressing the wrong button here because I'm doing it from my cell phone. So let me see.
I'm sorry I went a little long. So much to say.
No, but yes, no, but it was very interesting. So no, I don't see any more questions. So let me just thank you once again, Ajit. It was a very interesting talk. Thank you for joining us. We hope to see you in the symposium. And thank you, Patricia and the Earth Center, for hosting this webinar. And well, thank you for the audience and everybody else who has joined us today.
I will ping you later, Shubha.
Okay. Bye, Ajit. Bye, everybody.
Very much,
Thank
Ajit,
you.
and all the participants. It was a lovely session. Thank you, Patricia.
Bye.
Thank you.
Okay, thank you. So just, Shubha, are we having our meeting today? No?
Well, I was going to ask that because when I said anybody else coming, nobody else replied. But let's go there, and if there are more people, then we'll have
Okay.
it.
Okay. Let's leave here.
Okay, we